so thank you very much for giving me this opportunity to discuss about our recent research about spike protein. So already Tanaka-sensei today explained that what is spike protein. So I'd like to skip some of the slide because of this overlap. But anyway, the spike protein is very important protein for the entry of SARS-CoV-2 into the human cell. So we'd like to study this protein and to prevent the infection. So we have two interests in the study. So the one is the prediction of the conformational changes between the inactive and active forms. So as you see, the down conformation is inactive form, and active form, at least one RBD domain is up. So how this conformational change happen, that is great interest for us. And for this conformational change, what kind of interaction is important? And the second question is, what is the role of glycans in the spike protein? So as you see these figures, the right figures, glycan, the spike protein is on the surface. There are so many glycans. However, due to the flexibility of glycans on the surface of the spike protein, in cryo-EM studies, so these glycan structures are not clearly visualized. So in these figures, Wonpyo-Im, Lehigh University in US, so their teams built the glycans based on the mass spectroscopy data. So by using MD simulation, so we'd like to answer these two questions to some extent. So MD, we are using classical molecular dynamics simulations so to understand the dynamics aspects of spike protein. And for this, we are using and developing MD software Genesis. And this software has been developed in RIKEN mainly. And it allows the high performance MD simulations on parallel supercomputers like K, Fugaku, and other supercomputers. And we have freely distributed this software under LGPL license. And we have already spent more than 10 years from the initial start of the development, but still the development is underway. And the latest version, version 2.0, is actually optimized for Fugaku in the effort of the co-design by the Johnson, this person, and Kobayashi-san, in collaboration with Fujitsu and other RIKEN developers of Fugaku supercomputers. So both the this 2.0, this is a mistake. The latest version is 1.6. 1.6 and 2.0 beta are distributed from this website. So in this figure, we show the performance of Genesis on Fugaku and oak forest packs. So in both cases, the Genesis is quite scalable if we increase the number of computer nodes on Fugaku or oak forest packs. So in the first list performance, we almost reached to around the 60 nanosecond per day if we use 128 number of nodes on Fugaku supercomputers. So using Genesis, we are simulating spike protein on the surface of SARS-CoV-2 in solution. So we are in this simulation, we use Wonpyo's model of spike protein attached the glycans. So you can see the flexible dynamics of glycans on the surface of spike protein of the down form and active form. So using the simulation trajectory, we analyzed glycan structures on the stabilization of both down inactive and active up structures. And this figure is one of the examples of the snapshots from the targeted MD, starting from down conformation to the up form of spike protein. And here, 3 glycans touched at the 165 and 234 and 343 are highlighted because they are important for the stabilization of each form. The first, in the down form, the glycan attached the N165 interact with the RBD8 domain, and also the 343 glycan interact with RBD in the C chains. However, during this conformational change, the interaction partner is changed. Now, in the up form here, the glycan at the 343 loses its interaction partners, while the 234, so this one, is intruding into this cavity, so inside. So such information is nicely summarized in this cartoon. So you can see how differently glycan interacting with the down form and active form. So next, we'd like to answer the first question. So how we can simulate the large conformational change. So to visualize such complex motion, we are coarse-graining these structures using 27 Bs. So this coarse-grain representation was used for the spike protein in these papers to compare different spike protein in different viruses. So here, the spike protein structure is represented using the 27 Bs. However, if we use these 27 Bs and analyze the simulation trajectory using principal component analysis, so we can nicely visualize this down conformation and also the up conformation, and also transition simulation using targeted MD from down to up. So we consider this is a nice representation to see conformation dynamics. So now I'd like to show the movies using this coarse-grained model. So you can see the ribbon model of the protein, as well as these beads representing the center of mass of domains. However, you can see that the left movies, starting from down structure, stay in the down conformation. And also the up structure, if we start MD simulation from the active up form, it also stay again in the active form. And if we plot the fringe landscape of this simulation, you can see clearly these two distributions are separated. So we decided to use replica exchange method. It was the temperature replica exchange and Professor Okamoto developed more than 20 years ago. But now people prefer to use more the REST, REST simulation. And REST2 was invented by a different group. And in this case, solvent temperature is kept room temperature in all replicas, but the solute temperature is different, like replica exchange. And our modification we call GREST, Kamiya-san developed. And in this case, we can flexibly define the solute region. Now, the solute is not actual solute molecule, but a part of the solute molecule or a part of potential energy function. So this flexible definition allows us the much more opportunities to apply this method for very large system. So these simulations are the application to the protein ligand binding simulation. Here, not only the ligands, but also a part of the protein are selected as solute in that method. And we also apply this method to the protein conformational change in multi-domain proteins. And here, we selected solute as the Lennard-Jones and electrostatic interaction of surface charge residues. So here, this ribose binding protein, as we are using as example. And here we just selected the only few charged surface residues as solute region. So only 393 atoms are selected as solute. But because of this, you can see that much wider conformational regions are sampled in the hollow form and also the apo form compared to the conventional MD simulation. We can sample more states using this method, which we call GREST SSCR. So then we apply this method to the spike protein without glycan and with glycan. And this simulation started from down structure, but you can see that's more great motion in the RBD domain. And this also, same thing is true for the spike protein with glycans. And also, if we start from the up form with glycan, so not only the one up, but also the two up structure can be seen. So in this way, we simulated much wider conformational spaces using a new method, GREST SSCR. So we are now investigating the landscape and structures sampled in this simulation in more detail. So interestingly, recent cryo-EM structures not only the spike, but also bound with antibodies. So we can see not only one up, but also two up or three up structures. So this also suggests the great flexibility of this region in the spike protein. So by investigating such conformational flexibility using MD simulation, so we believe that we can obtain useful information for future development of anti-drug or anti-vaccine development. So that's the story today. And I really would like to thank these people who worked in this project. So thank you very much.